Our next topic is protein hydrolysis and denaturation. And this is looking at what sort of metabolic or destructive processes go on inside of our body with regards to protein. So the first one that we're going to talk about, protein hydrolysis. This is actually the reaction that takes place when we metabolize protein. So after we've eaten some sort of protein source, what our body actually does with the protein. Inside our body, we will break proteins apart, breaking the peptide bonds or the amide bonds. And this just completely disassembles the protein. It takes it apart, producing or leaving us with all of the individual amino acids that made up that protein. So it produces individual amino acids. Now, we don't break those down. The individual amino acids our body uses to put together and build new proteins. So again, we eat the protein. Inside our body breaks all the amide and peptide bonds inside that protein and turns that protein into thousands of individual amino acids. We don't break down the amino acids, but instead we take those amino acids and we assemble them to make new proteins. And that's what we're going to talk about in chapter 22. So we're going to just look at an example of protein hydrolysis, and I'm going to we have to draw a peptide for this. So I'm just going to draw a peptide that has 3 amino acids in it. It's going to be small, but that's just to try to keep it from taking up too much space in our notes. And you'll get the idea. Nitrogen, hydrogen, carbon, hydrogen, carbon, oxygen, and then this is the end. So just 3. And this is going to have, let's say our first R group is going to be a CH with 2 CH3s on it. So this is valine. And then our next one will be CH2OH. So that's S. And then our last one, CH2 COO minus, that's D. So this reaction actually, it's the opposite of assembling a peptide. So a couple sections ago when we were looking at taking 2 or more amino acids and putting them together to synthesize a peptide, we're doing the opposite in the hydrolysis reaction. I'm going to highlight the amide bonds or the peptide bonds, like the places where the individual amino acids are connected to each other. When we did this reaction in the other direction, when we were combining amino acids to synthesize a peptide or a protein, we saw that we lost a water molecule every time we created a peptide bond. So a water molecule was lost or produced for every peptide bond that was created. Now, in the reverse reaction, the hydrolysis reaction, we have to put those water molecules back. So we need a water molecule for every single peptide bond or amide bond that we're trying to break. So for this one, because we have 2 peptide bonds, we need 2 water molecules. And what we're going to end up getting out of this are just 3 individual amino acids. So I've circled the first one, and I'm going to draw exactly the components that are in the little peptide. And what I'm going to do is leave a little line here. So this is the bond that I'm breaking. And I'm going to draw the next the next guy over. So the second amino acid in the chain, I'm going to circle that, this in a different color. So now I'm drawing this guy, CH, C double bond O, CH2OH is our R group, and I'm going to put a little empty bond there where the peptide bond was, and then also on the other side as well. And so in this spot right here and right here, this is where we're going to put the equivalent of a water molecule. And it's going to go on, let's do it in a different color. We're going to get 1 oxygen on the C terminal, and we are going to get 2 hydrogens on the N terminal. And that's going to put our charges back on the oxygen, negative charge, and on the nitrogen, a positive charge. And we're going to continue this pattern for the third amino acid down here, this guy. So let's start by just drawing exactly what we have. Nitrogen with a hydrogen, carbon with a hydrogen, carbon-oxygen double bond with a single bond to an oxygen, CH2, CO, O-. And in the peptide bond, we're going to add a water molecule into this spot, and the way that we add it will be an oxygen on our carbon, on our C terminal, O-, and 2 hydrogens on the nitrogen to give us N+. And we don't need to do anything out here because it's already O-, so that was, you know, the end of the molecule anyways. And we also don't need to do anything out here because it was already NH3+ to begin with. So here are the products, these individual amino acids ready to be reassembled. Let's have a look at sapling. Homework 9, question 18, very similar to what we just did. Here's a little peptide, and it says modify the structure to show the amino acid hydrolysis products. So what we are going to do for this is break it apart into the individual amino acids. And this looks like there are 4 amino acids. So the first thing we have to do is find the location of those peptide bonds. Those peptide bonds are located between the carbon-oxygen double bond and the nitrogen. And that's kind of a bummer because it took the whole oxygen off. And then let's break the next one over. It looks like I've got the hang of it now. Oh, that was too much. So I've broken all of my peptide bonds, and now I need to add to my carbon-oxygen double bonds, I need to add another oxygen, and I need to do that for all of them. To the carbon-oxygen double bonds where I broke the bond, I also need to make sure that I'm adding charges on those oxygens. And then for the nitrogens, I need to add hydrogens, which I can do quickly with the plus hydrogen tool. I put hydrogens on those, and don't forget, I also need to put positive charges on those. And it looks like, ooh, these positive charges are covering up hydrogens. Remember, don't stress about that. They're still there. They're just being covered up. So that's all that this question is asking for you to do.